Approximately 69 million people a year sustain a TBI, but researchers have found that a peptide, CAQK, can specifically localize to the lesion site within 30 to 60 minutes to stop tissue damage in its tracks. Researchers have identified a breakthrough tetrapeptide known as CAQK. It is a homing peptide composed of 4 amino acids: cysteine, alanine, glutamine, and lysine. Its mechanism of action centers on its high affinity for a specific glycoprotein called tenascin, which is upregulated only at the site of the brain injury, the injured part of the brain, the site of the actual damage. When administered intravenously, CAQK acts as a molecular GPS, ignoring healthy regions, solely delivering the therapy exactly where the barrier is broken. Once the injury is located, the next step is to shut down the inflammatory fire. Once again, this content is for educational purposes only as it relates to laboratory settings involving legal animal models. In preclinical models of nerve injury, the peptide ARA290 has demonstrated a 23% improvement in neuropathic symptom scores compared to placebo groups. That's huge. One of the most potent modulators of brain inflammation is ARA290, also known as zimbinatide, derived from EPO, the substance that Lance Armstrong illegally administered to himself, meaning it doesn't just affect red blood cells. Instead, it binds to the innate repair receptor to trigger a cytoprotective signal. Research shows it can significantly reduce brain infarction volume, effectively reprogramming the tissue from a state of damage to a state of active repair. After cooling the inflammation, we have to restore the brain's electrical communication. This is educational content only. I do not promote personal use of any of these compounds for research into legal animal studies. This experimental peptide, PE22-28, is nearly 400 times more potent than its predecessor, allowing it to jumpstart brain communication with an inhibitory potency of just 0.12 nanomolar. To fix the post-traumatic decline in neuronal excitability, researchers developed PE-22-28. This synthetic peptide blocks the TREC1 potassium channel. After an injury, these channels often leak, causing neurons to become hyperpolarized and silent. By blocking this leak, PE-22-28 facilitates depolarization and restores hippocampal firing rates. This doesn't just wake the brain up, it enhances synaptic plasticity, helping the brain relearn lost functions and paving the way for new growth. Once again, this content is for educational purposes only, not medical advice, as it relates to animal models and animal studies only. Long-term treatment with the peptide P21 has been shown in preclinical models to increase the pool of newborn neurons in the hippocampus by a staggering 80%. That's massive. As the brain enters the repair phase, the compound P21 becomes the star. It mimics human ciliary factor to trigger neurogenesis. Its mechanism is unique. It inhibits the creation of scar-forming astrocytes and forces the brain to produce mature, thicker neurons instead, increasing dendritic spine density, which are the branches neurons use to talk to other neurons in simplistic terms. In TBI models, it successfully reversed the loss of dendritic density and reduced levels of hyperphosphorylated tau, which is the primary hallmark of CTE. But for these new neurons to survive, they need a stable source of energy. Again, this content is for educational purposes only as it relates to laboratory settings involving legal animal models. do not promote the personal use of any of these compounds. Neural cell death after a brain injury can reach 40%. However, targeted mitochondrial therapy with SS31 has been shown to slash that death rate down to just 19%, essentially in half. Recovery, when it comes to brain injury, is energy intensive, and that's where SS31, elamipretide, comes in. It is a first-in-class antioxidant that binds to cardioopsin in the inner mitochondrial membrane. By stabilizing the connection with cystochrome C, it prevents the catastrophic energy failure that usually kills neurons. In 2018 studies, it significantly reduced brain edema and improved motor function by protecting the cell's power plants from oxidative stress. Once energy is restored, we must safeguard the brain's long-term memory. Educational only. I do not endorse the personal use of any of these compounds, only as it relates to legal animal studies. A clinical study on TBI patients found that the peptide Pinealon reduced the average number of memory errors by 44%, bringing error counts down from 16.3 to just 9.1. The final phase of recovery is obviously epigenetic regulation. Pinealon is an ultra-short peptide that enters the cell nucleus to interact with histone proteins. By modulating signaling pathways, it prevents the brain from destroying dendritic spines, the branches that neurons use to communicate with other neurons, which are also the physical storage sites of your memories. Clinical scans show it improves bioelectrical brain activity and reduces memory-related errors by 44%, providing a long-term safeguard for the brain's new hardware. By integrating these targeted compounds, we can finally transform the future of neuro-recovery. My name is Salem Savage from Chem Center Bio. This is for educational purposes only, as it relates to laboratory settings involving legal animal models, never for personal use. I do not endorse the personal use of any of these compounds.